Investigating the adaptive behavioural plasticity of camouflage in crab spiders. Behavioural plasticity is the ability of an organism to modify its behaviour in response to changing environmental conditions. It's a flexible adaptation strategy that can play a critical role in survival, particularly for predators like crab spiders that rely on this process for successful prey capture. For crab spiders, their ability to blend seamlessly with flowers allows them to ambush unsuspecting prey. Through adaptive camouflage, these spiders are able to adjust their coloration and position, making them nearly invisible against various backgrounds. Scientists have been studying the behavior of crab spiders for years. Previous research has highlighted a few fascinating behaviors, like how these spiders select flowers that match their color morph, or even use scent-based or olfactory camouflage to stay hidden from prey. However, there's a notable gap in our understanding. While some aspects of crab spider behavior are well documented, we know surprisingly little about how these spiders might adapt to varying prey densities or floral types, which could provide new insight into their predatory strategies. Despite being known as powerful predators capable of subduing larger insects, we still don't fully understand how frequently they capture prey or the specific process of their ambush behavior. Additionally, the complex decision-making that goes into choosing specific backgrounds for camouflage remains underexplored. Background choice, while crucial to effective camouflage, is an underappreciated aspect that deserves more attention. Hence, future studies can focus on the mechanisms behind their predation behavior and how behavioral shifts may occur as a response to changing environmental conditions. This has significant relevance in the real world as climate change is continuously altering the environment. Rapid shifts in temperature, habitat structure, and food availability mean that species must adapt quickly to survive. Species with high behavioral plasticity, those that can adjust their behaviors in response to changing conditions, may have a distinct advantage in these dynamic environments. Our aim is to determine whether crab spiders can adapt their camouflage strategies to environments where prey availability is constantly changing. How does the variation in prey populations affect the camouflage-related concealing coloration behavior used by crab spiders as a predation strategy? This is an important area of study as it helps us understand how these predators respond to dynamic environments with fluctuating prey availability, and can help expand our limited knowledge on how the adaptive ability to alter behavior, such as optimizing hunting strategies, can maintain predation success and aid in the survival of crab spiders. In a controlled experiment setting, the prey population numbers were manipulated to examine how fluctuations in prey availability influence the crab spider's behavioral adaptations, particularly its camouflage strategy used to conceal itself while capturing prey. By adjusting prey densities, the study aimed to determine whether variations in prey abundance affect the spider's ability to modify its color-matching behavior to blend into its surroundings more effectively for successful ambush predation. The sample size consisted of a total of 32 individual crab spiders, which have a white coloration specifically adapted to camouflage amongst white tea tree flowers. This sample size allows for a sufficient sample population to account for any variations in data. The crab spiders were randomly obtained from various locations across a site at Smith's Lake, which is known for its high density of white tea trees. In addition, 14 black house ants were also collected from the same sites to serve as prey for the spiders to observe potential behavioral adaptations. Samples including both spiders and prey were collected from white tea trees using the beading method. This technique involved gently striking the branches with a stick to dislodge insects clinging to the leaves of flowers with a net positioned beneath to catch the falling insects. The insects were gathered into a test tube using a pooter. The spiders were then separated into individual plastic containers. The study was conducted in an enclosed test tube where one crab spider was placed inside alongside a small tree branch. Through random allocation, ants were then introduced, with the number of ants varying based on one of the following 4 experimental conditions. The control: crab spiders were placed among white tea tree flowers without any prey present. This allows us to assess how spiders behave naturally when there is no immediate opportunity for predation, providing a comparison point to identify any behavioral changes induced by the presence of prey. 3 treatment groups were designed based on prey availability, consisting of treatment 1. 2 black house ants were introduced, representing a scenario of limited prey to examine how the crab spiders adapt their hunting strategy when prey is scarce. Treatment 2. 4 ants were introduced, simulating a moderate level of prey availability. This provides insight into whether there is an optimal prey density that influences the spider's camouflage behavior. Treatment 3. 8 ants were provided, representing a scenario of abundant prey. This would provide insight into how a spider would change its camouflage strategy when there is abundant prey as opposed to a limited number. To quantify predation behavior and provide comparisons between treatment groups, 2 main types of camouflage predation behavior were identified, which included concealing coloration and flower mimicry. Concealing coloration is observed when a spider has its whole body settled amongst petals to conceal itself. Flower mimicry occurs when a spider positions itself near a flower with all arms outstretched in an attempt to mimicry the flower structure. The duration in minutes spent performing that behavior was recorded across all treatment groups, including the control. Through varying prey availability and observing the spider's predation responses, each of these treatment groups were designed to study how prey density influences key aspects of predation behavior in crab spiders. The varying ant numbers would ultimately help answer our research question and provide data that would help explain whether spiders adjust their strategies based on the relative availability of prey.